Greetings to all of you who are joining us for this set of learning series. And for the new members, the CSI Global Health Project hosts free seminars every 2 months. And the seminars happen on the last Thursday of the month, with speakers presenting a range of palliative care and rehabilitation related topics. And the series aims to optimize opportunities for local, national, and international networking within palliative care and capacity building for researchers. And today we are going to have Professor Maddocks lead the presentation, and he will present for 30 minutes and we'll have a generous 20 minutes for the question and answer series. A few housekeeping rules during the webinar. I'll request all members of the audience to mute themselves and kindly remain muted throughout the seminar. If you have any questions, please post them in the chat box and the technical team will be monitoring throughout. They will be written down and the speaker will respond to them after his presentation. The title of the series today is Clinical Trials in Palliative Care. And now I'll briefly tell you more about the speaker of the day. Matthew is an academic physiotherapist at the Cicely Saunders Institute, King's College London. He completed a PhD on the role of exercise in cancer cachexia in 2010 and has since held the National Institute of Health Research, NIHR, postdoctoral position, and also coordinates clinical trials and career development for fellowships. He is also a co-lead for the palliative and end-of-life care theme of the NIHR Applied Research Collaborative for South London. He leads a team undertaking clinical and health services research that advocates for rehabilitation and palliative care for people with serious illness through improved management of complex symptoms. And these include breathlessness, fatigue, weakness, and syndromes, including cachexia and psychopenia and frailty. So Matthew is a professor of health services research and rehabilitation. And we know that rehabilitation is one of the underfunded and least received services of palliative care. So Matthew is definitely an asset to the field. Over to you, Matthew.
Thanks, Eve. I'll just share the slides. Is this one showing okay?
Yeah.
Perfect. So I won't take longer than half an hour. And the title of the seminar is very broad today. And I was trying to decide whether to talk about particular studies that I've done or focus on a particular subfield in palliative care. But instead, I've taken a fairly methodological approach to the session. And so there should be a lot of content around basic trial design and the description of clinical trials, especially randomized controlled trials. Some of this for people may be very familiar. And in that case, hopefully it's just reinforcing what you know, and we can build on the content of the slides and the presentation with some discussion after. And for other people, hopefully it's a useful introduction to randomized controlled trials. So it's fairly basic, and I've tried to keep the content just to the essential components of trial design. And of course, after all of this, there's a lot of detail that teams of people designing and delivering clinical trials will work through together and make decisions about. But lots of it is based on this kind of framework. And the initial question is, I guess, why we need clinical trials or why we need randomized controlled trials. And we'll all appreciate that in our field or fields of palliative care and rehabilitation, and often in our settings of health services and health systems in our countries, there's an absence, at least a relative absence, of high quality evidence to guide decision making. Maybe we could make a comparison with oncology, which as a specialty is about the same age as palliative care, but through lots of rigorous trial design and of course lots of industry support and funding, the oncology field has developed a very strong and robust evidence base. And we see the effects of that because treatments for cancers across most cancer types are improving quite rapidly. They're developing quickly, they're becoming more personalized and tailored. And lots of that is driven by a very high quality, rigorous evidence base, which is principally born from randomized controlled trials. The trials are also good because without that evidence, we have lots of variation in practice. And people are either doing what their seniors do within their centre, or they're borrowing good practice or borrowing practice from other centres, or they're following very basic guidance that's coming from recommendations. So this variation in practice is unfair because it means that some people will end up getting very good practice and other people will get substandard or imperfect practice in their centre. So through evidence and through randomized controlled trials or clinical trials, we're able to try and reduce that variation in practice so that everyone gets the best care possible. And then the last two bullet points go hand in hand. And again, with this strong evidence base, we're hopefully improving the care that can be offered to people. And that will help prevent people being exposed to unhelpful or ineffective treatments. And it will also reduce harm in a broad sense because it means that we understand which treatments and how treatment should be delivered to patients and their families, so that more people in our population, more of society get access and they get a receipt of effective treatments and effective palliative care practice, effective components of palliative care or rehabilitation or any health service for that matter. And lots of people will be familiar with this evidence pyramid, and for a good reason. Randomized controlled trials have historically and continue to be quite high up this evidence pyramid. The randomization means that two people or each person in the trial has an equal chance of going into one group or another. And what that does is balance out the groups in the clinical trial for characteristics that we know about and that we can measure, but also characteristics which we were unaware of or haven't measured. And these we would call confounders. Even if we don't know about them, they're randomly distributed and they're by chance fairly allocated between one group or another. The model has recently been changed, and on the right hand side you can see a kind of more contemporary way of looking at the evidence base with respect to trial study design. And again, the randomized controlled trial sits quite high up the top, but you can appreciate that some trials are better than others, and of course a well-designed cohort study, for example, may provide equally strong evidence. And I like here also that the magnifying glass, the lens or the way that we're looking at the evidence would represent a systematic review or meta-analysis, which is another type of study design. So a clinical trial has been defined for many decades now. It's a form of planned experiment. So it's an experiment, and that means we change or we interfere with what's going to happen to that person, of course, when they're consenting to take part in the clinical trial. The patient, or sometimes groups of patients, are the experimental units. So this is what we would put into one group or another. The term subject some people would have used historically, and now people would much prefer participants. They're participants in these clinical trials. But it's that person or that group of people who are being interfered with insofar as we're changing the natural course of what might happen to them. And we're interfering by experimenting and offering an intervention. And the other defining feature of a clinical trial is it's designed to find out the most appropriate treatments for future patients in a similar condition, with a given condition or in a particular context. So these are experimental study designs, and they're not descriptive study designs. And because of that, because we're experimenting and making interference with the care of someone taking part or consenting to a trial, they're all prospective. That means we recruit people and follow them over time. The randomized controlled trial is conducted under controlled conditions, and we're making measurements and observations of what care people are receiving. Unbiased is quite a strong word. They perhaps have a low level of bias. So through the rigorous methods which we're using in clinical trials, we're reducing different biases. For example, we're reducing a selection bias, which is picking certain groups of people to be studied. We're reducing an allocation bias or a preference bias, where people tend to receive what they want to, or the clinician tends to offer what they think is best for that particular person. So we're reducing this bias, and in doing so, getting closer to a truth, if we believe or accept that there's a universal truth. And then finally, the clinical trial should be large enough. And there are some rules of thumb where people will say that this was a small trial or this was a large trial. When they do that, actually in the pain literature, a small trial has less than 200 people in each group. And there's a kind of, it's connected to some statistics. But when we describe a trial as being small or large, it's very arbitrary, really. It's taking a number and just making a value based on that number. When we say large enough, we should be talking about how the trial is designed to detect a certain level of difference or certain effect from a treatment. And we would use the word powered. It's an adequately powered or sufficiently powered trial rather than a large clinical trial. So for all clinical trials, there's a very nice framework, and this is probably the one thing to write down or take away, if anything, from today. We can use this PICO or PCOT with a T framework. And when we want to describe a clinical trial which we're conducting or designing, or indeed we've read and we want to tell other people about, the PICO framework is a very useful way to describe the trial. And it's relating to the population, that's the people taking part in the trial, the intervention, which is the therapy or the treatment or the service that's being studied, the control, and the control may be a usual care, or it may be a different intervention, or it may be the current practice for that particular condition being studied. And then finally, the O is the outcome. And in a clinical trial, really related to its design, the primary outcome, which is usually one clinical outcome only, is the most important one. We'll measure other things, but the design of the trial is around the primary single outcome measure. And then the timing gives a feature of when that outcome will be assessed. So we may, for example, and we've got some later in the presentation, have a trial which is, let's say, early palliative care, which is the intervention, compared to usual care, the control, in people with advanced cancer, the population, and the effects on quality of life at 2 months would be the outcome and the timing for that particular example. So for each of those in turn, the population is describing those patients or those family members or the participants of the study, the people that are going to be approached to take part, consent to take part, and then offer their time and information and provide their data for the clinical trial. And we would describe the eligibility criteria using inclusion criteria and exclusion criteria. And usually we would do that in this order. So the inclusion criteria will describe the patients by, for example, their condition or their stage or their age or the particular group they're belonging to and who we want to approach within the study. The inclusion criteria is usually about the people taking part. Sometimes people will include a feature of the setting or the clinical context or where people will be recruited from. And for me, this is most useful to come after the inclusion criteria, because if we're only including people based on a particular setting or place where their care may be delivered, then in some ways we might be making that population quite narrow. And we're reducing how useful that particular trial is to other people with the same condition, but being seen in a different setting or a different context. And then the exclusion criteria. Some people have very, very long lists of exclusion criteria, and that can lead to difficulties getting people who are eligible for your study. There should be a very valid reason for every single exclusion criteria. And often it should relate to the treatment which is being studied, if there's like a safety risk or a contraindication to that treatment. Sometimes it's if the people are already receiving that treatment which is being studied. So if we were doing a palliative care service trial, we wouldn't want to be recruiting those people who are already in the service. And then also there's some exclusion criteria which would relate to their ability to take part and understand and contribute useful data to the trial. And this might be things around their cognitive function, if people have got impairments with cognition, and also their language. And that's their ability to understand what's involved and consent appropriately, and then also complete and engage in the treatment and the outcome measures taking part. And a really important part of a consideration for the eligibility criteria is, of course, if you're able to deliver what you said you were going to do. We could all design a perfectly sophisticated and grand trial, but if we can't deliver that, if we can't recruit people, then it's a kind of all a bit made up and it's all in theory. So we want to very carefully consider those eligibility criteria and understand, ideally collecting some data to support that case, that we'll be able to recruit this group of people from the settings which are available to us. And we'll be able to follow those people and make measurements on them and have them take part in the trial. So moving to the I, so we've done the P and now the I, the intervention. For lots of trials, in a very broad sense, the majority are undertaken to do with medicines or drugs. We call CTIMPs, a controlled trial of an investigational medicinal product. But in palliative care and actually in rehabilitation, we're not often studying drugs, and we're studying other interventions. These could be very simple or discrete treatments. For example, some techniques to help people with their breathing, or a frame or some equipment to help people with their mobility. They may be things like packages of communication training or education to patients. They may be things around services which are focused around a particular symptom, or palliative care services as a whole. And some examples here from a kind of African context is a trial of dignity therapy. So this would be a particular therapy being studied. An educational intervention to support people with pain and to help them self-manage pain. And then at the bottom there, palliative care, a service level intervention. Now, a drug in many ways is quite simple, but as we move towards the things which we're typically studying in palliative care, we're talking about complex interventions. And complex interventions involve lots of different parts, and they're offered in different ways to different people. So if you imagine your usual practice in palliative care, as a clinician, as a team, you're holding lots of skills, and you've got a kind of a toolkit of things that you might offer. And if one person comes in and they've got lots of physical distress related to uncontrolled pain, then what you might offer them would be different to a next person who's coming in who has a horrible crisis and family situation, very emotional distress, lots of spiritual issues and concerns that they're wanting help with. So the service is designed and should be offering different things to different people. So in the next slides, we'll talk about this framework, which comes from the Medical Research Council. And this is offering a structure with which to start to build up the study of those kind of services, those complex interventions. This is the oldest, and it's been superseded twice, we'll see. And the idea here was that if, as in the drug world and in the medicinal product landscape, we can successfully get things through from preclinical up to phase 1, 2, 3, and 4, and the approval, the marketing would take place after phase 3. The regulators would have a phase 3 trial. They'd allow people to offer that drug and get people to purchase and commission it, and the phase 4 is the marketing phase. And they were saying in the same way we should try and be able to do that for all interventions, including the very complicated ones. And initially, the black text on the top, they're saying the equivalent of this phases of drug trials is developing the theory, modeling and exploring, which is working out which parts of the service work best and how to get it in the best state possible to study. And then in phase 3, this definitive word that you might hear people say, definitive trial, this is the real testing of the intervention. And then ideally it's successful, and then we talk about the long-term implementation. But this is a very idealistic way, it's a very optimistic way of looking at complex intervention testing. And many people never get past the exploratory stage, and many people do their trial and they find that the design wasn't quite right, and they don't, or the trial wasn't big enough, and they don't find the effect. So there's been 2 further iterations, and you can see this and the next one, which I'll go to now, are far less linear. There's an understanding that we don't necessarily go from designing, feasibility testing, full randomized trial, and then implementation. There's a lot of back and forth, and there's a lot of swapping between one approach and the next one. And the important feature of the most recent framework from the Medical Research Council is the core elements. And this is about the real understanding of that intervention and how important the context is, where it's being offered, what else is around, the involvement of stakeholders and the people involved in delivering, receiving, commissioning, working alongside the intervention being studied. And then also lots around the theory and the understanding and the writing down of how that service is being organized and being offered, and how it's bringing about the intended effect. So this would be a good reference point for people to seek if they want more information. Then the next is the control. So we've got this intervention which we're interested in, and we've hopefully, if it's a complex intervention, spent the appropriate time building it up. Now we want to test it against something. And a very important point about trials is you're comparing, it sounds very straightforward, but you're comparing one group to another. And what you're interested in is creating a gap, creating a distance between what people in your treatment group receive, typically, and what people in your control group receive typically. So usual care and an intervention, if this, for example, is a palliative care service, you want to try and make sure there's a difference between those things. And there's this famous trials that they call TEML trial in the US setting that had a lot of gap between usual care and palliative care. It was testing early palliative care. And in that time, in that context, usual care was just oncology services, and you wouldn't see anyone with palliative care expertise. Whereas now, in the US at least, usual care involves quite a lot of palliative care involvement or consideration, and a lot of generalists who are trained in spotting concerns and offering a little bit more treatment. So the gap between palliative care and usual care now, at least in the lots of high income settings, is being reduced. And we can also have other comparators like a control which is offering people some form of attention. And in depending on some treatments, we can have like a placebo, which is like a sham or a mock-up of the treatment, but it's not the real thing wanting to be tested. And then finally, we have the outcomes. And I mentioned before, but the primary outcome, the measure, the part of people's functioning, their quality of life or their function or the target symptom is the most important one. And we can include secondary measures, things that we think might change alongside that in our studies. But the kind of standard advice is not to include too many secondary outcome measures, because we're not really designing a trial to test those things. The design is really centered around the primary outcome, the thing we want to test if we can change and influence with our experiment. And then people might use this term endpoint, which includes an element of time. So it might be pain after 8 weeks, quality of life at 3 months, or physical function after 6 months. These sorts of things would be a primary endpoint. And there's a whole other conversation about the types of measures we would choose to understand those. Now, just a couple of slides on the sample size. And the figure here is not designed to kind of frighten people, but it's just to show these calculators that you can find on the web, and they're usually freely available, and with a little bit of support from people, you can work through them and understand your sample size. What they're really doing is just drawing a line from somewhere on this left-hand side where it says the standardized difference, and that's the amount of effect you're expecting to see in your trial, or you're designed to be able to observe. And then a line from that left-hand side to the right-hand side, the power, which is going from 0.995 at the top to 0.05 at the bottom. And all you'd be doing, my mouse may be showing, is drawing a line, let's say from 0.4 here, and we're going across to 0.8 power. And where it crosses this middle axis is the number that the sample size calculator will give you. And it's slightly different depending on whether your significance level, your p-value, is 0.05 or 0.01. So that line, if we were to draw it going somewhere here, would fall around here, 170 people, if we're setting the significance level at 0.05. So it's not that fancy in the background. It's just a fairly straightforward calculation. But what you're telling it is how much power you want in your study and how big a difference you think you'll be able to observe in your study. And again, there's a bit more information than people may need, but what you're doing with that sample size is reducing the amount of error. And if you've got a bigger sample size, what you're really doing is reducing the number of times, if you're doing several studies, or the chance, if you're doing one study, of making an error in your observation. And you can have a type 1 error, which is when you say the treatment worked, or there was a difference between the two groups, when in truth, there wasn't. You found a positive in your trial, but it's false. And this is related to your p-value. And usually, because we'll often have a p-value of 0.05, this is really meaning about 5% of the time, we'll have a statistically significant finding, and the truth is that there wasn't really that order of difference between the treatment and the control group. But far more common is the type 2 error, which is when you make an observation in your trial, you don't observe a difference, so you say the treatment isn't effective, or we didn't find the level of difference that we were looking for, when the truth is that there is a difference, there is an effect, it's just slightly smaller than that trial was designed to do. And although we have a very rigorous way of setting our p-value at 0.05, and we're happy that 5% of the time we're saying something works and in truth it didn't, our p-value, our power, apologies, is usually set at 0.8 or 80% power. And what this is really meaning, what we're accepting with that, is to say that 20% of the time, 1 in 5 studies almost, they determined that there wasn't a difference or there wasn't an effect from the treatment, whereas in reality, in truth, there was. The trial was underpowered and the sample size in that trial wasn't big enough. So when we say a small or a large trial, it's really relating to this issue of the amount of difference we're hoping to observe and whether or not we've recruited enough people to do that. So it's, as I say, in principle, a larger trial, a larger clinical trial is reducing the error that we're going to make. And if we find a difference, we can believe it. And if we find that there wasn't a difference, we can believe it. If we've got a trial that's too small, we might find things just by chance. And then finally, we've got a couple of key features of these designs. So the randomization, this giving people a very equal chance to receive one thing or another, will reduce the bias in that study design, and it will reduce the confounding in that study design. So a bias is skewing things in one direction or another, away from the truth. And of course, we want to be centered on the truth. And confounding is something which we're not aware of affecting what we observe in that trial, and misunderstanding something like cause and effect. And if we use very simple randomization, and again, you can get this on the web, you could get predetermined lists which are random, then we'll be doing all of that allocation to one group or another by chance. If we're using things like a record number, the day of the week, alternate assignment, treatment control, or someone's date of birth, then although we might think this is introducing some chance, it's not random in its nature. And it's possible to start to influence the allocation, or it's possible to start to understand where the next person might go. If everyone on a Monday or a Wednesday in our clinic gets randomly allocated to the treatment, then this isn't random, and this is a very predetermined sequence which everyone would understand, patients, clinicians, people undertaking the trial. So just go for simple randomization from the web or from a list of random numbers, and don't use things like case numbers or date of birth and believe that that's a chance allocation. And then the other feature of clinical trials is blinding. And so people will sometimes talk about single-blinded trials or double-blinded trials. And very usually, a single-blinded trial is that the participant doesn't know which treatment they received. And again, in a drug trial, this is quite plausible and possible because you're taking a tablet and you're not sure if that's a real tablet or a placebo one. But in a service or in lots of physical interventions, this is quite tricky to achieve. And in many of our trials, just by nature of what we're studying, it's not possible to blind participants. And in that case, what we should be doing is thinking, who else can we blind? Who else doesn't need to know about the treatment group allocation? And this can be the people who are making the assessments, most of all. The people who are assessing the outcomes of that treatment at the end of the trial can, if possible, and if possible, they should be blind, and they shouldn't know whether or not the next person they're making an assessment of received the treatment studied or the usual care. And then the last 3 slides, just to finish, is the different types of clinical trial or randomized clinical trial. So we have what we'd call a parallel trial, where we're randomly allocating one person to either the treatment group or the control group. The example I've given here is that TEML study. So these were people with newly diagnosed advanced lung cancer. And in that study, they either got randomly allocated to a control group, usual care, which was just oncology, or they got a treatment group, which was oncology plus specialist palliative care involvement. And this trial was famous because it had an improvement in quality of life, and it was also, you may remember, one of the first ones to observe a difference in survival of patients, which they thought might be related to the more appropriate use of aggressive cancer treatments towards the end of life. And in some trials, you do have more than 2 groups. You can have a control and maybe 2 different treatments which you're studying. People like that as an idea, but in doing so, you're at least halving the cost again, increasing the cost by half. And although it may seem like efficient to do this grand trial with lots of treatment arms, it's usually just, in real terms, more expensive. It can sometimes feel like you're not quite sure what it is you're trying to test, so you're just adding in different groups. So for many good reasons, just a two group parallel trial is conventional, and it is kind of most efficient in terms of the energy spent. So the next one is a cluster trial, where instead of randomly allocating individuals, you're forced to randomly allocate groups of individuals. This is really necessary when your intervention involves upskilling staff or training staff. Because you can imagine, if you go to a group of clinicians and you're giving them some new skills or some new available techniques to treat something or to provide better care in an aspect of someone's life, then it's not really plausible that they can use those skills, remember them for some people and forget them and not use them for other people. Just practically, if you're giving someone, in this example, the skills and a plan to better advance, to provide better advanced care planning, you can't really say do that for this person but not the next person. So in that case, you give some clinicians the skills and other people not, and the groups of patients who are seeing those clinicians are the things that were randomly allocated. And then finally, we have the crossover trial. So here, instead of one phase of random allocation, what you're randomly allocating is the order of different treatments. So this is particularly useful for medicine trials, and you can use it in some things. Like I found an example from Eduardo Barrera, which was people sitting or standing to deliver bad news. And they were rating how compassionate that care was when the physician was standing up or sitting down delivering some bad news about a cancer progression. But they only work when there's a short acting effect of an intervention and when it doesn't have any carryover or long-term effects. Because what you're assuming is that between this point here of the first phase and the second phase of making measurements and observations, you're getting what we would call washout, and people are returning to where they were before randomization. If that doesn't happen, then you get a carryover effect, and you're not really comparing like with like. So you're not meeting the assumptions of this particular trial design. So it can be very useful with short-acting interventions. And because people act as their own controls, there's a very small sample size requirement. But that said, they're very difficult when people are have progressive disease and they're changing quite rapidly, because they're not the same person from here to here. And they don't work in long-acting drugs where there needs to be some time to build up the effect. And they also don't work when there's a carryover effect from drugs. So there's an example here of oxygen that's provided in hypoxemia, which was a while ago, and people may find other ones. So I'll finish here. There's a very brief summary. I think the main message is that the clinical trial is a really useful, strong design, but you need lots of careful planning. And I would encourage people to take advice from methodologists and people with experience in trials, because you only get one chance to do the design. And after that point, everything is invested, and you're spending your time and money and energy from everyone delivering that design. This PICO or PICOT framework is very useful to start thinking about the core aspects. And then the sample size is based on your primary outcome. And you may need to use this crossover or a cluster design, depending on the question you're asking and the intervention that you're testing. And then finally, these complex interventions, if we're thinking about testing a service or testing a whole approach, then there are distinct phases for testing, and they would include lots of questions about feasibility before we make that large investment to test the effect. Yeah. So I'll stop there, and hopefully we'll have a good time for some questions and maybe some discussion. Thank you.
Thank you very much, Matthew. And now the floor is open for questions and answers. Jane, you have the floor, please.
Matthew, thank you so much for your presentation. I hope, can you hear me?
Yeah.
Yes.
Okay,
yes.
Great. I have a question about the concept of feasibility. I don't know that I understand the word correctly in a sort of clinical trials understanding.
Yeah.
I guess because my sort of lay use of the word feasibility is this possible? And, you know, obviously possible could mean, you know, cost effective or, you know, practically possible or whatever. What's the sort of understanding of feasibility in a clinical trial setting? I'd like to understand a bit more about that. Thank you.
Yeah, thanks. So I guess first it's quite recent where there's a bit more consistency in people understanding or using that term. And people used to say pilot, I'm just piloting this. And now a pilot trial, in general, people are saying a pilot trial, you've decided what the trial is going to look like, and you're just doing that first delivery of it. Now, feasibility is when you do a feasibility trial to try and answer some questions and uncertainties about your trial design. There are several aspects to feasibility, and you need to spend time thinking which ones are most important. Some of it is about recruitment. Can I recruit this population? And can I get them, support them through the trial? And so most commonly, I think, the feasibility trial is saying our feasibility outcomes is, can we recruit 40 patients within a year, or can we recruit 1 in every 4 patients that we approach about this trial, and can we follow those patients through the trial and make the intended outcome measures on them. There's also some feasibility questions about the intervention delivery.
Leave the positive out.
A bit of feedback, I think.
The mental, so you know how to know what a filter is.
Irene, please mute yourself. Thank you.
Yeah. So some questions are about the feasibility of delivering your intervention. And here the questions are, here's what we intend to deliver. How much of that can we deliver to those people in our intervention group? And many feasibility trials don't have to be randomized. And then the final set can be the feasibility of making measurements on those participants taking part in the trial. So, for example, someone actually yesterday, I was examining someone. They'd done an exercise study for people with myeloma having treatment. So their feasibility questions: can we recruit from that patient population? Can we deliver the intervention, in this case exercise, and how much of what we hope to deliver can we? And then finally, can we make measurements that we would use as the primary outcome? So a lot of people, they would say, I'm doing a feasibility trial, but they don't spend the time really clearing up what it is they have uncertainties around. And then related to that, if you're not sure if you can randomly allocate people to one treatment or another, then your feasibility trial could be randomized. But if you're saying, I'm not sure if this population would agree to being randomly allocated to one treatment or a control, then it's very appropriate to have a randomized aspect to your feasibility trial. But if you've not really got a lot of uncertainty about that, and your feasibility questions is more about, can we deliver this approach as an intervention, then there's really more value in having an uncontrolled feasibility trial, because you're getting more exposure to understanding how your treatment can be delivered or which outcomes can be measured. So for many people, they're tempted to do a randomized feasibility trial, but if you've not really got uncertainty about that ability to randomize people or ability to recruit people to be randomized, then in some ways you're not wasting, but you're not getting the best value out of every participant, which is where people sometimes have a randomized feasibility design, but every 2 people get an intervention for 1 person that gets a control. So I hope that's a little bit clearer.
Kennedy?
Yes, thank you, Eve. Thanks, Matt, for that presentation, really informative. Before I ask my question, let me just comment about the feasibility. I think Matt has given quite a good explanation in terms of what it's all about. Let me just say that actually for the map care project, which we are doing, is part of the feasibility. So we were asking the questions, can we recruit elderly people with serious but morbid illness who are 50 years and above? That is what we are asking across 3 countries. And also, can we follow them up or retain them for the next 4 months? So those are the questions that we're trying to answer in terms of feasibility. But I think Matt has also explained that in detail. Yeah, thank you. So my question was a little about the outcomes. Of course, you know that actually when you are trying to come up with a sample size, you base on the primary outcome. But there are some studies that they have like more than one primary outcome. So in terms of determining sample size, do you choose which one should be used for the primary outcome, or maybe you need to use both of them, depending on how many outcomes you have?
Yeah. Yeah, if people genuinely have 2 primary outcomes, then the convention is that they have to set their p-value at 0.025 rather than 0.5, because in a way, and then especially with the regulators, and I think that's good practice to follow, a success is only if both of your primary outcomes change. And often you're doing this to provide evidence of some kind of objective improvement in an outcome, and also some patient-reported improvement. and you could have a co-primary. Other people, if they can't quite pick one, then they'll say, oh, we might have a composite primary outcome, which means you can have a selection of 2 or 3, and if one of them improves, then that will contribute to your overall interpretation. But again, I think especially with larger funders, you are going against convention, and in doing so, you'll be carefully looked at, and they'll wonder why the team can't make a decision about one outcome or another, or they'll wonder why you need to do a composite outcome. And they're not in palliative care, but there are some examples, maybe like in cardiology. They have a composite primary outcome that would include hospitalization and death. And there are cases where they say the treatment worked on the basis of there being fewer adverse outcomes, even though there were more deaths and fewer hospitalizations relative to the control group. So there are some ways where the composite outcome is not providing the best clarity. And in fact, when people are having a co-primary outcome, at least in medicine trials or in drug trials, it's usually stipulated and it's a requirement of the regulator rather than a choice of the investigators, because you should then be very strict and use your p-value across those 2 outcomes. So you should really set your p-value at a higher threshold, because you don't want your chance to be increased 5% plus 5%. You still want to keep to the convention of having a 5% chance of a false positive.
Thank you, Matthew.
Yeah, very tricky. Lots of discussion about outcomes, and
Yeah.
usually it's a group decision, and I suspect most of the time there's a kind of majority vote rather than unanimous consent.
Thank you, Matt. Thank you.
Thank you. Richard? Richard, you have your hand up.
Yeah, thanks, Steve. Thank you, Matt, very, very much. I enjoyed that. Can I ask if you could just another moment or two on the standardized difference, because I think that was probably the most technical bit of the presentation when you showed that really helpful slide for the sample size calculator. Could you just run through the terms again, standardized difference and power, just so that's as clear as possible, please?
Just trying to find one. So a standardized difference is a way of normalizing, like comparing different things on the same scale. And a standardized difference, which people would also say effect size, the same thing, is ranging from 0 to 1, and it's based on the change in populations and how varied one population is to another. And it's I'm just trying to see if I can find you can visualize, you're trying to compare 2 groups of people and how separate those people are. You're not comparing the average person in each of those groups, you're comparing the group as a whole. So a standardized mean difference, what that means is it's standardized to a distribution within your population, and you're comparing one group to another using this scale from 0 to 1, which is it's a bit tricky because it's related to the standard deviation, which is another measure of how people are spread out. But there are different standardized mean differences, and we would be able to say this is a small difference, this is a moderate difference, this is a large difference. And usually a small one is like 0.3, a large one is 0.5, a medium is 0.5, and a large one is 0.7. And what that means is that the medium one, for example, moderate, is 0.5 half of a standard deviation, which is a spread of the population. But it's all about how much gap is there between one group and another. And the bigger the number, the more gap there is between those two groups. And if you're looking for a very big gap, you don't need as many people in your study to observe that. And if you're looking for a very small gap, you need more and more people as the gap gets smaller. And the mistake that many people make while they're thinking about, can we really deliver this trial, they be a little bit optimistic about how effective their treatment might be. And they start to think, okay, in what we're going to see with this service is a large effect size, a large standardized mean difference, let's say 0.8 or 1 even, and that gives them a very attractive sample size. The calculator says, okay, now you need 24 people in each group to do your trial. But what you've said that, in doing that, you've said, I think the average person in my treatment group is going to fundamentally change in where they sit in the population. I'm going to really change this measure for them for the better and create such a regular common difference between one person in my treatment group and the next person in my control group. And in doing so, if the reality is that the treatment is effective, but not quite as effective as you hoped it would be, because you were all along thinking about this nice small trial that you could do, then what you've done is reduce the power of your study and you've increased the chance of your type 2 error. So you do that underpowered trial, you didn't find the difference, so you've got a negative, but it was underpowered, and so really you've got a false negative. And if we do too many small optimistic trials, then what we'll be doing is populating the literature with trials saying this service is ineffective, this service didn't lead to a difference in the outcome for the patient, which we overcome in some ways when we can do like a meta-analysis, which is why it's important people are using sensible measures in their trials. But if we do lots of those small trials, and if we're too ambitious or too optimistic in our sample size calculations, and we're thinking about best case scenario, then what you're really doing is saying, I'm going to increase the chance of a false negative finding. So I'll try and find a resource because there's a bit more to it.
Brilliant.
But that's
Fantastic.
the consequence,
So
yeah.
If I could simplify that for my brain, what you're saying is don't over anticipate the effects that your intervention will have, because if you think it's going to have a great big effect and you don't recruit many people, you'll end up not detecting anything at all and then missing any effect that you did have.
Yeah.
Brilliant. Thank you so much for doing that.
Thank you so much, Matt, and thank you everyone who has been in attendance. A kind reminder to please complete the survey and also to send through the list of participants to Kennedy. Richard, I know you have a meeting which should be starting shortly.
Correct.
Unfortunately, we will not take on any more questions, and we look forward to our next session in 2023. Thank you very much, everyone, and have a wonderful rest of the day.
Thanks all. Thank you.
Yeah.
Thanks.
Please feel free to leave at leisure.